Welcome to another regular dose of pain. The first leg of parasitology starts with protozoan parasites, the chum bucket of all single-celled organisms, eukaryotes particularly. We should expect a lot of the structures that we would see in your typical eukaryote cell. The nucleus, and some may have a macro and micronucleus, the cytoplasm, there's the cell membrane, other pertinent organelles and structures like mitochondria, the Golgi apparatus, ribosomes, and the locomotory structures. These are some of the things that you'll see across the different protozoan groups. How could a single-celled organism be so diverse in appearance? The diversity really comes from the variations and the modifications in these same structures. From a diagnostic standpoint, we only need to focus on the structures that are key to species identification. When you're being asked to label structures, focus on the structures that will help you identify that this organism is indeed whatever it is that you ID it to be.
Oh, ito, cytoplasm. Oh, ito, cytoplasm. O ito, cytoplasm. Kailangan ulit-ulitin pa ba? Hindi na po.
As we've mentioned, there's not only a diversity in the form, but there's also a diversity in the rest of their life history patterns, like their mode of feeding. Protozoans can be holozoic, which means they eat like animals. Ingest, then they digest, and then they egest. Orophycal. Holophytic, they feed like plants. So they actually have photosynthetic organelles. Euglena. Walang kamatayang euglena. Saprozoic protozoans absorb their nutrition via diffusion. You might also see this being defined as osmotrophy. Coprozoic protozoans each gladly mixotrophic protozoans. Walang pili-pili. They employ multiple modes of feeding. After you've eaten your meal and you've had your fill, the ultimate purpose of life, to be able to reproduce. Protozoans are able to do that in many different ways as well. For asexual reproduction, protozoans can split into two, so there's binary fission. There's also multiple fission. The genetic material, the nuclei, are the ones that really split off first. But it still remains within one cell membrane, just floating in the cytoplasm. And then there comes a time when everything just pinches off, and then you now have a bunch of multiple organisms. You may also hear this termed as schizogony or merogony, especially as we approach the group of apicomplexans. For sexual reproduction, there's conjugation. As in, whatever happens there, they exchange genetic material. A lot of these protozoan parasites, you'll see in their life cycles that they're able to employ both asexual and sexual modes of reproduction. Protozoan parasites. You'll find them in two life stages. The trophozoite stage. Troph means eat. Zoid means animal. The motile stage, the one that's moving around, bopping around, eating. And then there is the cyst stage. This is like the I am stressed, do not disturb me. I'm gonna hide in an armor of resistance to pain and suffering. That's the cyst stage. And why a lot of protozoans have this? Remember, you're a single cell. And what is the prerequisite for almost, if not all life on Earth? Water. If you don't have a life stage that will allow you to survive periods of just extreme dryness, and you're just this flippity floppity thing that will almost easily just you're gonna die. When the trophozoite form transforms into the cyst form, it is said to encyst. When the cyst form transforms into the trophozoite, parang nagising siya, it is said to exist. I exist, therefore I exist. Now on to the medically important protozoan parasites. Here they are. Shit! Dami, ilan yan? Oh my god, so many names. Chylomastix, Retortamonas, Giardia, Diantamoeba, Entamoeba, Plasmodium, Toxoplasma, Cryptosporidium, Gregorina. If you're going to be doing that, like just memorizing them one by one, this is the organism, this is the life cycle, this is the mode of transmission, this is you're gonna have a hard time. Instead of committing each and every single species to memory, let's try to find out their common characteristics and group them as such. This is when the systematics of organisms becomes very helpful. That's the whole point of systematic. Scientists in the early days, they didn't have the high-tech microscopes and the DNA sequencing technology that we enjoy today. The systematics of the parasites that you see here was based on the structures that were visible using the tools that they had at that time. So they did what they could with the basic light microscope. This partly explains why protozoans were traditionally classified based on their locomotory structures. Kasi yun lang naman yung nakikita nila sa microscope. Flagella, cilia, pseudopodia, the apical complex. While it's not really a locomotory structure, it's more of a cell invasion structure.
Actually, hindi kita yung apical complex sa light microscope. It's really locomotory structures.
Morphological based classification can still be very useful even in the advent of phylogenetics because phenotype is a reflection of the genotype. More often than not, this also sort of reflects that you sort of share the common ancestor. Kasi parang namana nyo yung structures na yun, di ba? Yung lolo ko may flagella, kaya ako rin may flagella. there are cases where two similarly looking organisms actually have a very distant relationship. They don't really share any recent common ancestors. Conversely, you could have two morphologically dissimilar organisms, but if you trace the lineage, they do share a very recent common ancestor. This is the whole point of phylogenetic systematics. We want to know who's related to whom, not just based on external morphology, but also really based on genetic similarity. You have this table. Let's turn it into a tree. The tree shows the major clades. You'll see that they're called supergroups. This sort of shows you how each supergroup may be possibly related to another and how the subgroups in each supergroups are somewhat related to each other. And because this is still an active area of research, it still has elements of traditional classification. You might notice from the old grouping versus the new grouping, they're still actually quite similar. They kind of just changed the names and shuffled a few members around, but some of the groupings here are still really based on the morphological similarities because they have yet to establish the ancestral or the genetic aspect of things. So please, expect changes! Dahil hindi ko siya natiis, isama na lang muna natin siya. Say hi to everyone. For the purposes of this classification, let's just follow this tree that you can find in your book. So if we actually take a closer look at this tree, most of the protists, yes baby, most of the protozoan parasites that we'll be discussing have been scattered across these different eukaryotic supergroups. For the parasites that we'll be studying, they're actually spread across the groups Excavata, Chromalveolara, and Amoebozoa. And because protozoan systematics is still very much under constant revision, let's have a look at this tree. Compared to the tree in your book, ano yung pinaka nawala? The supergroup Chromalveolara is gone.
Surprise, motherfucker!
The former members of Chromalveolata, the Straminopiles, and the alveolates, together they were lumped with the supergroup Rhizaria, and they are now called SAR. Ano ba naman yan? Kakaaral ko lang nung isang tree. Ipapaaral mo na naman to. Pabago-bago naman eh. Hold up, I just want to emphasize that things will be constantly changing. And for protozoans, this will be uncomfortably frequent. Please don't be surprised if after a few years, this tree will be somewhat outdated. For the moment, that's kind of where we are at. Siguro may nagbago na nga as of the time I'm recording this, tapos hindi ko lang talaga na-update. Kasi, tamad. In the next doses of PaIN, we will discuss the protozoan parasites one major group at a time. Para hindi sumabog yung utak nyo. Since our objective in the laboratory is for you to be able to identify these parasites, mainly using light microscopy, I leave you with a few tips on how to identify them. Oh my god! Nainip na siya. Ang tagal-tagal ko na raw nagre-record dito. Diagnostic sample. What did they give you? Did they give you shit on a slide, blood on a slide, sputum on a slide? Is it a tissue sample, like a biopsy, like a sliver of liver? Sliver of liver! Gulat siya. Because a diagnostic sample can more or less tell you which parasites you're going to be expecting. Morphology. With light microscopy, that is perhaps the best way to confirm what parasite you have because it's like, yan yung itsura niya. That's why it's a lot more satisfying to identify parasites microscopically. With DNA, you just see a bunch of bands. Oh, okay, nagmatch na yung bands. It's Giardia. Whereas, pag nakita mo yung mukha ng lolo, wow, I found the Giardia in this piece of explosive diarrhea. With morphology, a few things to take note of. Locomotory structures. Do you see flagella? Do you see pseudopodia? Do you see cilia? The nucleus. How many are there? Is there a big one and a small one? In terms of the features of the nucleus, look at the nucleolus. Is the nucleolus right smack in the center of the nucleus, or is it off to the side? Other unique structures, the axostyle, undulating membrane, the kinetoplast. Some of these are actually unique to a specific organism or a specific group of parasites. Life stage. Are you looking at a cyst stage or a trophozoite stage? There are some parasites that don't have a cyst stage. Size matters. It really does. Diba? I'm not lying. Size matters. Ba iniisip nyo, when you're looking at it in the microscope, what magnification are you using? Oil immersion? You're probably looking for a protozoan. If you're using like high power or low power or scanner, God forbid, if you can see it with your naked eye, you're probably looking at something bigger, like worms. Also in terms of the actual measurements, like how many microns is this thing? So that'll also help you figure out if you're looking at maybe a worm egg, protozoan, piece of plant debris. At least, yan yung sa lab. The lecture part, which will help you also with the lab, is in terms of pathogenesis, symptoms, patient history. Saan nanggaling yung pasyente? Ano yung kinain niya? Nilagnat ba siya? Nagtatai ba siya? Nagsusuka? May dugo ba yung plema? Having that information, which you will learn from the lecture, and then you put that in with what you see in front of you, that'll help you identify the parasite. So this doesn't only apply to protozoans, but for the rest of the organisms that we will encounter in your next doses of PaIN. I'll see you in your next dose of PaIN, where we will discuss the excavates.